Have you ever thought about what it would be like to read the actual instruction manual for life? I mean, what if we could decode the very blueprint that tells a flower how to bloom, or what makes you, you? Well, that's exactly what we're going to talk about today as we jump into this incredible world where biology meets massive, massive amounts of data. So, to really get why this field even exists, we have to take a quick trip back in time. Think of it like a tale of two labs, two completely different eras of scientific discovery. And trust me, the difference between them is just mind-boggling. Okay, look at this. On one hand, you've got Gregor Mendel, the father of genetics, back in the 1860s. He spent 3 years on a single experiment with his pea plants. And after all that time, his results, they could be summed up in a single paragraph. One person could easily look at that and see the patterns. Now, fast forward to today, a similar experiment, it might take just a few hours. But the data it spits out, we're talking terabytes of information. That's more data than you could fit on thousands of laptops. And that incredible leap forward, well, it created a brand new problem. We got so good at generating data that we started, quite literally, drowning in it. It's an ocean of information so vast that no single person, not even a huge team of people, could ever hope to sort through it all by hand. To give you a sense of the scale here, just look at this number. 3.2 billion. That is how many letters, the A's, T's, C's, and G's, are in the genetic code, the genome, of one single human being. It's a number that's almost too big to even wrap your head around.
And this really puts it into perspective. If you were to do nothing else, 24/7, but just read that sequence of letters out loud, it would take you 4 months. And that's just reading it. That's not even beginning to try and understand what any of it actually means.
This cartoon just nails the feeling, doesn't it? It's like someone hands you a jigsaw puzzle with over 3 billion pieces, but there's no picture on the box to show you what you're building. I mean, where would you even start? It's this exact challenge that forced scientists to come up with a totally new way of thinking. So how in the world do you solve an impossible puzzle like that? Well, you bring in a partner that's really, really good at finding patterns in enormous amounts of data. You team up biology with the incredible power of a supercomputer. And this amazing partnership has a name, BioInformatics. It's not just one thing. It's this beautiful fusion, right? It's where biology, computer science, and statistics all come together to tackle this massive data problem head-on. You can think of it as standing on 3 pillars. First, you've got biology. That's where the questions come from, and it's where we get the raw data. Then there's computer science. This gives us the tools, the software and the hardware to store and process all this information. And finally, you have statistics. This is the magic that lets us find the truly meaningful patterns and separate them from all the random noise. Okay, so we know what it is, but how does it actually work? To get that, we need a super quick refresher on the basics of how genetic information flows inside our cells. It's kind of like learning the basic grammar before you try to read the whole book of life. Everything starts with DNA. The easiest way to think about DNA is as the master blueprint for a building, or maybe a giant cookbook with every recipe you could ever need. It holds every single instruction for building and running an entire living thing, and it's kept safe inside the cell's main office, the nucleus. But here's the thing. A blueprint can't build a house by itself, right? You need workers. And in our cells, those workers are proteins. They're the ones who actually read the instructions from the DNA and then go out and do all the work, building things, carrying messages, basically making everything happen. And this whole flow of information has a name. It's called the central dogma of biology. It's pretty simple, really. The master blueprint, the DNA, is way too important to leave the main office. So a temporary copy of one specific instruction is made. Think of it like a photocopy. That little message travels out to the factory floor of the cell, where it's read or translated, and the workers build the exact protein that's needed for the job. So now we've got all this code, billions and billions of letters. How do we even begin to find the important parts? How do we find the specific instructions that actually matter? Well, the main method is something we all do every single day without even thinking about it. We learn things by doing them over and over, sure, but we also learn by comparing stuff. You know that a chair is a chair because you've seen a bunch of them and compared them. One might be wood, one might be metal, but they share some core features that your brain recognizes. It's all about comparison. And that is the absolute core of bioinformatics. We teach computers to do that exact same thing, but on a massive scale. The number one technique is comparison. By comparing a new genetic sequence to millions of others that we already know about, we can start to see patterns, similarities, and differences that actually mean something. So the basic workflow looks a little something like this. First, a machine reads the raw DNA, but it can only read it in tiny little pieces. So step 2 is this huge computer puzzle, taking millions of those short fragments and assembling them back into one long correct sequence. And then finally, the most important part, we take that new sequence and compare it against a massive global database of every sequence we've ever seen before. Here's probably the simplest way to think about it. Imagine you have 2 copies of a 1,000-page book, and you know there are a few words changed between them. Trying to find those changes by hand would take you forever, right? But a computer program? It could scan both books and highlight every single difference in seconds. That's pretty much what we're doing with DNA. So, why? Why go through all this trouble? Why compare billions of letters of genetic code? Because hidden in those tiny differences, in those patterns, are the answers to some of the biggest questions in medicine and biology. By comparing the genome of a healthy person with someone who has a genetic disease, we can find the exact little typo in the code that's causing the problem. And once we know the cause, we can start to design new medicines to fix it. It helps us understand evolution, to see how we're all connected. At the end of the day, this has a massive, direct impact on improving real human lives. This isn't just theory. This is changing the world. The amazing thing is we're really just at the beginning of this adventure. For the first time in human history, we have the ability to read life's source code. The real question is, as we get better and faster at reading and understanding this book of life, what incredible secrets are we going to unlock next?